Hello everyone, welcome to Subhash Chandra Tutorials. Today I am going to discuss the topic mitosis. First we have to understand the meaning of word mitosis. Mitos means thread, osis means state. Mitosis, during the process of mitosis, spindle fiber formation takes place from the centriole, hence the procedure is known as mitosis. The process of mitosis in case of plant cells is explained by scientist Strasberg, and the same process of mitosis in case of animal cells is explained by scientist Fleming. The term mitosis is coined by scientist Fleming. During mitosis, the parental cell gives rise to two daughter cells containing same number of chromosomes as parent cell. If parent cell containing 2n chromosomes, then daughter cells produced through mitosis will be containing same number of chromosomes as parent cell. The process of mitosis takes place in case of somatic cell and in case of some germinal cells also. The somatic cell may be haploid or may be diploid. Haploid number of chromosomes are represented by n. Diploid number of chromosomes will be represented by 2n. Basic chromosome number will be represented by X. We know the process of spermatogenesis and oogenesis. The process of spermatogenesis or oogenesis contains mainly three stages. They are multiplication stage, growth stage, and maturation stage. During maturation stage, the process of meiosis takes place, but during multiplication stage, the multiplication of a germinal cell takes place through the process of mitosis. Based on the morphology of a nucleus and the chromosome, the process of mitosis can be classified into 5 stages, namely interphase, prophase, metaphase, anaphase, and telophase. The interphase is further classified on the basis of DNA replication into G1 phase, S phase, and G2 phase. Chromosomal material during interphase is fully extended and uncoiled. Some of the early cytologists called interphase as a resting stage, but actually the RNA, protein, and DNA replication takes place during interphase of a cell division. Based on DNA replication, interphase can be further classified into 3 substages. They are gap 1 phase, synthesis phase, and gap 2 phase. It constitutes 25 to 50% of an interphase. RNA and protein synthesis takes place during G1 phase. Ribosomes are produced in the nucleoli of a cell, and then they migrate towards outer membrane of a nucleus and get attached to the outer membrane, and also some of the ribosomes attached to the outer membrane of an endoplasmic reticulum leads to rough endoplasmic reticulum where protein synthesis takes place. S phase of an interphase, during which synthesis of a DNA takes place. I mean, replication of DNA takes place during S phase of interphase. RNA and protein synthesis takes place at very slow rates during S phase, but histone proteins required for structure of a chromosome are synthesized during S phase. G2 phase means gap 2 phase, where considerable RNA and protein synthesis takes place, and some components of microtubules are also synthesized during the G2 phase. mRNA is also synthesized during G2 phase. This is a structure of a cell where we can see nucleus having nuclear envelope and chromosome as the chromatin fibers, nucleoli, and centriole. Genetic material of a cell is replicated during synthesis phase of interphase, and which helps in formation of spindle fibers also get duplicated During S phase. During prophase, chromosomes appear as thin chromatin fibers. As prophase progresses, condensation of a chromatin fiber results in formation of a chromosome. This is the structure of chromosome. Chromosome contains centromere and contains arms. These arms are called as chromatid. Before S phase of an interphase, the structure of a chromosome contains single chromatid. As a result of DNA replication, this arm splits longitudinally, results in two sister chromatids. This part of a chromosome is known as kinetochore. The condensation of a chromosome during prophase is mainly due to condensing. During mid-prophase, as a result of condensation, two sister chromatids of chromosomes are clearly visible. At the end of a prophase, chromosomes are short and thicker. In prophase, two sister chromosomes are coiled with each other. This is called as relational coiling. The relational coiling is of two types. They are plectonymic coiling and paranymic coiling. In plectonymic coiling, two sister chromatids of a chromosomes are coiled each other in such a manner like rope, where two threads of ropes are coiled around each other. In order to separate two sister chromatids, we need to rotate the chromosome to separate two sister chromatids. Plectonymic coiling is seen during mitotic prophase, but in case of meiotic prophase, we see paranymic coiling. What is paranymic coiling? Paranymic coiling means one sister chromatid is slightly slipped over another sister chromatid. There is no need of rotation of a chromosome to separate two sister chromatids. During metaphase, disappearance of a nucleolus takes place and the nuclear envelope disintegrate and the components of a nucleolus, components of nuclear membrane get attached to the endoplasmic reticulum. And during metaphase, the centrioles results in synthesis of spindle fibers. There are mainly two types in spindle fibers. Shorter spindle fibers called aster rays, the longer spindle fibers. In longer spindle fibers, they are classified into two: continuous spindle fibers and chromosomal spindle fibers. And the chromosomal spindle fibers are also called as discontinuous spindle fibers. Discontinuity of chromosomal spindle fibers is due to centromere region of a chromosome. And in case of continuous spindle fibers, there is no discontinuity due to chromosomes. During late prophase, the centriole synthesizes the spindle fibers. These spindle fibers get attached to the centromere region of a chromosome. As cell division progresses, as cell enters the metaphase, stretching of this larger part of a spindle fiber takes place. This results in placing the chromosomes at equatorial plate. This larger part of a spindle fiber starts to contract. This results in position of this chromosome at equatorial plate. During metaphase state, the centromere of all chromosomes are placed at equatorial plate. Metaphase, centromere of chromosomes are aligned at equatorial plate. As I explained, chromosomes are placed at equatorial plate, but during anaphase, the chromosomes split longitudinally at the centromere region as a result of contraction of a spindle fibers from each poles. As a result of longitudinal splitting of a chromosome, the interzonal fiber formation takes place between splitted centromere of a chromosome. As a result of splitting, two sister chromatids starts to move away from each other towards poles. As we all know that there are mainly four types of chromosomes based on the morphology of a chromosome, that is based on situation of a centromere in the chromosome. They are metacentric, submetacentric, acrocentric and telocentric chromosomes. In case of metacentric chromosomes, the centromere is present exactly at the center of a chromosome. In case of submetacentric chromosome, the chromosome consists of centromere slightly away from the center. And in case of acrocentric chromosome, the centromere is slightly present at the end of the arm, but not exactly at the end of the arm of a chromosome. In case of telocentric chromosome, the centromere is exactly present at the end of the arm of a chromosome. During anaphase, various chromosomes appears different shapes due to forces that are pulling the chromosome from equatorial plate to the poles. In case of metacentric chromosome, the chromosomes attains shape of English alphabet K. In case of submetacentric chromosome, chromosome attains English alphabet L shape. And in case of acrocentric chromosomes, the chromosomes attains J shape. And in case of telocentric chromosome, it attains I shape. The splitted chromosome are pulled towards poles mainly due to three forces. That is due to contraction of a spindle fiber. This results in movement of a chromosome away from equatorial plate to the polar region. And this movement is also due to stretching of a interzonal fiber. As I told that the interzonal fiber is formed whenever the centromere region of a chromosome is splits. Then 30 ATP molecules are required for movement of one chromosome from the equatorial plate to polar region. During telophase, the chromosomes starts to uncoil due to decondensation, results in formation of chromatin fiber. And also nucleolus starts to reappear and nuclear membrane also starts to reorganize around the chromosomal material at two opposite poles. At the end of telophase, cell plate formation begins. Cytokinesis. Cytokinesis means is division of a cytoplasm of a cell. The membranous elements of endoplasmic reticulum and the products of Golgi bodies get deposited at the center of a cell at equatorial plate, which results in formation of a cell wall and subsequently middle lamella. Cytokinesis starts at the center of a cell or at equatorial plate in case of plant cells, but in case of animal cells, invagination of a plasma lemma from all sides starts and meets at the center. This results in division of a cytoplasm into two parts. At the beginning of mitosis, we have a single cell. At the end of telophase, we have two daughter cells with same genetic material as present in case of parental cell.